Hi students, in the last class we discussed about the glycolysis pathway, right? That glycolysis pathway connected with the link reaction. The link reaction connected to the Tca cycle. So what I am telling, between the glycolysis and the Tca cycle, there is a connecting reaction is there. The connecting reaction is called as link reaction. Link reaction done by the pyruvate dehydrogenase enzyme. Link reaction done by the pyruvate dehydrogenase enzyme. That one is the membrane bound enzyme, especially present within the mitochondria membrane. So within mitochondria membrane there is a pyruvate dehydrogenase enzyme is present. That dehydrogenase enzyme involved in the conversion of pyruvate to acetyl CoA. Pyruvate to the acetyl CoA. So here the pyruvate molecule, 3 carbon content compounds, especially 2 pyruvate, end of the glycolysis process, 2 pyruvate molecules we have, right? The 2 pyruvate molecules involved in the production of 2 acetyl CoA molecules. So in this process, the 3 carbon compound converted into the 2 carbon compound. The 3 carbon compound converted into the 3 carbon compound. Very simple. Here the pyruvate structure is CH3 C double bond O, right? COO minus. So during this process, during the conversion, CH3 C double bond O, right? Yes, CoA. This is the structure of the acetyl CoA. Here coenzyme A is added. Coenzyme A is added. So this is the 2 carbon content compounds. This is the 3 carbon content compounds. Here carbon dioxide is removed. Here carbon dioxide is removed. So the pyruvate is converted into acetyl CoA. Okay. Here pyruvate dehydrogenase is the key enzyme. This is the multi-subunit enzyme. That enzyme requires thymine pyrophosphate, right? One of that vitamin substance it requires, and magnesium ions are required, and lipoic acid is required, right? Coenzyme A is required. Coenzyme A is required. So here these are the requirements for the pyruvate dehydrogenase enzyme, right? So the pyruvate is converted into acetyl CoA. Here thymine pyrophosphate is required, magnesium ions are required, lipoic acid is required, coenzyme A is required, right? See, during this process, carbon dioxide is removed. Carbon dioxide is removed. See, decarboxylation. The step is nothing but the decarboxylation. And next one is NAD is converted into the NADH plus H plus. NADH plus H plus. So this is nothing but oxidation. This is nothing but the oxidation. So this step is called as oxidative decarboxylation. This step is called as oxidative decarboxylation. So here oxidation is occurs, decarboxylation is occurs. Removal of carbon dioxide is nothing but decarboxylation. So this step is called as oxidative decarboxylation. So here the pyruvate is converted into acetyl CoA, right? So this reaction is called as link reaction. So within the link reaction, one NADH plus H plus production takes place. One NADH plus H plus production takes place. All of you remember the acetyl CoA which is interact with the oxaloacetate. So this is the 4 carbon compound. This is the 4 carbon compound interact with the 2 carbon compound. Here citrate is produced. The citrate is the 3 carbon compound. The citrate is the 3 carbon compound. So here acetyl CoA involved in the Tca cycle by the condensation process. The acetyl CoA condensed with the oxaloacetate. Okay. Here the citrate synthase is the enzyme. Citrate synthase is the enzyme. That enzyme involved in the production of citrate. So from here onwards, several steps are take place within the Tca cycle. So finally, within the Tca cycle, carbon dioxide production, water molecule production takes place, right? So this is about the link reaction. Next one we will discuss about the Tca cycle. This is about the link reaction. Next one we will discuss about the Tca cycle, right? So within that link reaction, pyruvate is converted into acetyl CoA, right? Coming to the next one is the Tca cycle. We know after the link reaction, the next cycle is the Tca cycle. Tca cycle indicates here tricarboxylic acid cycle. Tca cycle is nothing but tricarboxylic acid cycle. Tricarboxylic acid cycle. Sir, why this one is called as tricarboxylic acid cycle? Whatever that first formed, this is the citrate and succinate and isocitrate. These are the 3 compounds, right? See, the 3 compounds contains three compounds contains three carboxyl group. Here three carboxyl groups are present. Here three carboxyl groups are present. Here three carboxyl groups are present. So the very first three compounds contains the three carboxyl groups. That's the reason why this cycle is called as tricarboxylic acid cycle. Tricarboxylic acid cycle. So here Tca, right? That's the reason why this cycle is called as Tca cycle. So due to the first three compounds, okay, the first three compounds here carboxyl group contain compounds. That's the reason why this cycle is called as tricarboxylic acid cycle. See, the Tca cycle first identified by the Sir Hans Krebs. Name of the scientist is the Sir Hans Krebs. Remember, Sir is the respect word, Hans Krebs is the scientist name. That's the reason why the Tca cycle is also called Krebs cycle. The Tca cycle is also called Krebs cycle. All of you remember, I already said here this is one of the respected word, right? Sir, why he respected? Because he identified this cycle by the usage of the radiolabeled substances, especially carbon radiolabeled substances are utilized, and he identifies this Tca cycle and he got the Nobel Prize. Okay, that's the reason why before his name the Sir is added. The Sir is added, right? So that's the reason why his name is the Sir Hans Krebs, right? He got the Nobel Prize for the invention of the Tca cycle or identification of the Tca cycle, right? So that is about the Tca cycle. Then what is the use of Tca cycle? We know that oxidation of glucose molecules in that way or in that pathways. The very first one is the glycolysis of that link reaction. The next one is the Tca cycle. So oxidation of glucose molecules done by the Tca cycle, because we know that respiration process, especially cellular respiration process, within that cellular respiration process, the glucose molecules are finally converted into carbon dioxide and the water. Sir, in the glycolysis pathway, there is no carbon dioxide production. Then, how the glucose molecules are converted into carbon dioxide? Remember, within the Tca cycle, see, this is one of the carbon dioxide releasing step, and this is another carbon dioxide releasing step. So within the Tca cycle, carbon dioxide production takes place. Within the Tca cycle, carbon dioxide production takes place. And remember, see what I'm telling. Here carbon dioxide is produced, water molecules are produced. Then what is the use? High energy compounds are produced. That high energy compounds which are interact with that electron transport chain produces the ATP. So the concept is the cellular respiration majorly done by the three stages. One is the glycolysis, the next one is that Tca cycle, the last one is the electron transport chain. So now we are discussing about the Tca cycle. See, by the link reaction acetyl-CoA production, the pyruvate is converted into the acetyl-CoA, right? The acetyl-CoA condensed with the oxaloacetate. Oxaloacetate. So here oxaloacetate is one of the product of the Tca cycle. That one is condensed with that acetyl-CoA. Here coenzyme A released out. The coenzyme A released out. Okay, after that condensation process, here coenzyme A released out. Then these two are combined, produces the citrate. Remember, here condensation of oxaloacetate and acetyl-CoA done by the citrate synthase enzyme. Condensation of oxaloacetate and acetyl-CoA done by the citrate synthase enzyme. See, the citrate is the 6 carbon content compound. So oxaloacetate is 4 carbon content compound. Acetyl-CoA is the 2 carbon content compound. So oxaloacetate and acetyl-CoA, both these are condensed, which produces that 6 carbon compound, right? See, here coenzyme A released out. This reaction is called as condensation reaction. The very first step of Tca cycle is the condensation, right? Here, that citrate again, here the next step is the citrate is converted into cis-aconitate. By the removal of water molecule. So this step is called as dehydration. See, this reaction carried out by the aconitase enzyme. Remember, the enzyme name is aconitase. Okay, product is the aconitate. The product is the aconitate. So citrate is converted into cis aconitate by the action of aconitase enzyme. One water molecule is removed. Right. This step is called as dehydration. Removal of water molecule is called as dehydration. Next one is the cis aconitate converted into isocitrate. The cis aconitate is converted into isocitrate. See, by the addition of water molecule. By the addition of water molecule. See, here water, whatever that H plus and OH minus ions, whatever that H plus, that is nothing, water molecule is nothing but H plus and OH minus ions. See, removing from one location. Again the H plus and OH minus, adding at different positions. See, at particular positions, hydrogen atom and OH minus, okay, H plus ion and OH minus ions are deleted from the citrate. Then it produces the cis aconitate. Again, the same water molecule is added. The same water molecule is added, but the binding of H plus and OH minus ions occurs at different positions. Then the cis aconitate produces the isocitrate. The cis aconitate produces the isocitrate. See, both these reactions, removal of water molecules, okay, addition of water molecules, both these are done by the aconitase enzyme. So one step is the dehydration and the other step is the hydration. Addition of water molecule is nothing but hydration reaction. Addition of water molecule is nothing but the hydration reaction. The next step is the isocitrate is there. See, I like to explain here the number of carbon atoms. See here, 6 carbon compound. This one is also 6 carbon compound. This one is also 6 carbon compound, because here there is no removal of carbon molecules. That's the reason why 6 carbon compound, 6 carbon compound, 6 carbon compound. Right? See, the next one is that isocitrate is converted into oxalosuccinate. Isocitrate is converted into oxalosuccinate. In this process, NAD plus is converted into NADH plus H plus. NAD plus is converted into NADH plus H plus. See, this reaction is called as oxidation. We know removal of hydrogen atoms is nothing but the oxidation reaction. So the enzyme is isocitrate dehydrogenase. Remember, see, the reaction is the the reaction is dehydrogenation, means removal of water molecule. The enzyme name is isocitrate dehydrogenase. The enzyme name is the isocitrate dehydrogenase. So here oxalosuccinate is there. Oxalosuccinate again converted into the alpha ketoglutarate. Oxalosuccinate is converted into alpha ketoglutarate by the removal of carbon molecule. By the removal of carbon dioxide. Carbon molecule means here carbon dioxide. See, whatever that oxalosuccinate is there, oxalosuccinate is the 6 carbon content compound. Then the 6 carbon content compound converted into the 5 carbon content compound, because one carbon dioxide molecule is removed, one carbon molecule is removed here. That's the reason why the 6 carbon compound is converted into 5 carbon compound. See, removal of carbon dioxide is nothing but the decarboxylation. Removal of carbon dioxide, the reaction is the decarboxylation. Name of the enzyme is the decarboxylase. Name of the enzyme is the decarboxylase, right? So here there is a decarboxylase enzyme involved in the removal of carbon dioxide. Then the 6 carbon compound converted into 5 carbon compound. So alpha ketoglutarate, right? See, this is one of the decarboxylation reaction, right? The next one is alpha ketoglutarate. Alpha ketoglutarate is converted into the succinyl CoA. Alpha ketoglutarate is converted into the succinyl CoA. In this process, removal of carbon dioxide takes place. That is nothing but decarboxylation, right? See, other step is the NAD is converted into the NADH plus H plus. So this one is called as, see, this reaction is called as oxidation. This reaction is called as oxidation. So this reaction is called as decarboxylation. Total the reaction is called as oxidative decarboxylation. Total this reaction is called as oxidative decarboxylation. By the oxidative decarboxylation process, NAD is converted into NADH plus H plus and carbon dioxide removed, right? So here requirements are thymine pyrophosphate, TPP, thymine pyrophosphate, that is one of the vitamin component, right? And coenzyme A is required, magnesium ions are required. Remember, in this step also magnesium ions are required, in this step also magnesium ions are required, because both these enzymes are requires magnesium atoms for the activation of the enzyme. For the activation of the enzyme, this step and this step, okay, both these steps are requires the magnesium ions, right? So here NAD is converted into NADH plus H plus, here carbon dioxide removed, right? So here the 5 carbon compound converted into the 4 carbon compound. The 5 carbon compound converted into the 4 carbon compound, right? So here succinyl CoA production is there. That succinyl CoA, because here coenzyme A is added, that's the reason why this one is succinyl CoA. So here dehydrogenase, alpha ketoglutarate dehydrogenase enzyme, alpha ketoglutarate dehydrogenase enzyme, right? Here succinyl CoA converted into the succinate, succinyl CoA converted into the succinate. See, alpha ketoglutarate is converted into the succinyl CoA by the addition of the coenzyme A, right? The succinyl CoA converted into the succinate. See, this step is called as substrate level phosphorylation. See, this is the oxidative phosphorylation, this is the decarboxylation, this is the oxidation, right? Hydration, dehydration, condensation. So here this is the order, this is the 7th reaction. Here substrate level phosphorylation takes place. One of the phosphate molecule is taken by that succinyl CoA and coenzyme A is released out. The CoA is released out. The coenzyme A released out. Then the phosphate is added to that GDP molecules. Then GDP synthesis takes place. Okay, this reaction is called as substrate level phosphorylation. This reaction is called as substrate level phosphorylation. Here enzyme is the succinate thiokinase. Succinate thiokinase. So succinate thiokinase is the enzyme involved in the conversion of succinyl CoA to succinate, right? After that conversion process, remember here here GDP synthesis takes place. The GDP is equals to ATP. The GDP is equals to the ATP, right? The succinate, this is the succinate. See, this is the 4 carbon compound. This one is also 4 carbon compound. This one is also 4 carbon compound. The succinate converted into the fumarate. The succinate converted into the fumarate by the action of succinate dehydrogenase enzyme. By the action of succinate dehydrogenase enzyme, the FAD is converted into the FADH2. The FAD is converted into the FADH2. The reaction is the oxidation reaction. Removal of hydrogen atoms. That is nothing but the oxidation reaction. Okay, enzyme name is succinate dehydrogenase. So FAD is converted into FADH2. So here the fumarate production takes place. The succinate is converted into the fumarate. This one is also 4 carbon compound. This one is also 4 carbon compound, right? The fumarate by the addition of water molecule. Okay, here presence of fumarase enzyme. Presence of fumarase enzyme. That one is converted into the malate. The fumarate is converted into the malate. The fumarate is converted into malate. Presence of fumarase enzyme. Addition of water molecule will be takes place. So addition of water is nothing but hydration. This is the 9th reaction. That one is the hydration. Then the malate is converted into the oxaloacetate. Malate is converted into the oxaloacetate. This is one of the oxidation reaction. Removal of hydrogen atoms. Right? Oxidation reaction. So here enzyme is malate dehydrogenase. The enzyme name is malate dehydrogenase enzyme, right? The malate is converted into the oxaloacetate, right? So we know about from this onwards, because oxaloacetate combined with the acetyl CoA coenzyme A released out produces the citrate. So this is the Tca cycle. Totally here 10 steps are present. Within these steps, some steps liberates that carbon dioxide, some steps liberates that water molecules, and some steps liberates that NADH plus H plus, and some steps liberates FADH2. Okay, in certain cases GTP production, right? So these are the steps which are seen in the Tca cycle. So here I like to ask here, okay, how many number of NADH plus H plus are produced in one Tca cycle? How many number of NADH plus H plus are produced in one Tca cycle? This is the first NADH plus H plus, right? So this is the second NADH plus H plus. This is the third NADH plus H plus. So one Tca cycle, 3 NADH plus H plus are produced. 3 NADH plus H plus are produced. Okay, one Tca cycle, right? In Tca cycle, how many FADH2s are produced? Oh, only one, right? In one Tca cycle, one FADH2 production takes place. in one Tca cycle, how many GDPs are produced? Only one GDP production takes place. Only one GDP production takes place. So this is about that energy level for one Tca cycle. This is about energy level for one Tca cycle. See, I will explain the energy levels, but this is okay. We have to remember in one Tca cycle, 3 NADH plus H plus 1 FADH2, 1 GDP production takes place. Remember how many number of acetyl CoAs are produced. See, end of the glycolysis, 2 pyruvate molecules are produced. The 2 pyruvates are converted into 2 acetyl CoAs. The 2 acetyl CoAs involved in the 2 Tca cycles. This is the one Tca cycle. Okay, another Tca cycle also takes place because by one glucose molecule, 2 acetyl CoAs are produced. By one glucose molecule, 2 acetyl CoAs are produced. So here 2 into 3, 2 into 2. Yeah, 2 into 1, 2 into 1. So here what I am telling here, 2 Tca cycles are occurs for one cycle. Okay, 3 NADH plus H plus, 1 FADH2 for one cycle, 1 GTP for one cycle. So for one glucose molecules, how many Tca cycles occurs? 2. So that's the reason why 2 into 3, 2 into 1, 2 into 2. So in the next class we will discuss about how many number of ATPs are produced for one Tca cycle, right? For complete oxidation of one glucose molecule, okay, how many number of ATPs are produced from glycolysis to Tca cycle, we will discuss, right? That is about that Tca cycle. Remember, in all these steps, oxidative decarboxylation step is very important. Oxidative decarboxylation step is very important. So here substrate level phosphorylation, the step is succinyl CoA to the succinate, right? See, that is about the Tca cycle.